Hey, everybody, thanks for coming in. We're going to get started in a minute once everyone trickles into the webinar room. All right, great. Well, hello and welcome, everybody. Thank you for joining Southern Model Organisms Corp., SMOC for short, for our first webinar of summer 2026. If this is your first time meeting us, SMOC has 26 years of experience in providing genetically modified rodent models, covering custom gene editing services and over 18,000 ready-to-use models, including more than 1,000 humanized therapeutic target mice. Built on our expertise in genetically humanized animal work, we provide a comprehensive suite of full-service CRO offerings in addition to our fully humanized antibody development platform, which will be the topic of today's webinar, entitled SmocMab, a mechanism-aware platform for next-generation therapeutic antibody discovery. I'm Dr. Alex Sercel, a specialist in mitochondrial bioenergetics and director of West Coast Business Development here at SMOC, and I'll be today's host and panel discussion moderator. I'm joined by our panelists, SMOC's Chief Business Officer, Dr. James Jin, our Senior Vice President of Antibody Discovery, Dr. Lei Chen, and our Chief Executive Officer, Dr. Dongxiao Feng. It is my great pleasure to introduce today's speaker, Dr. Ningning Ge. Dr. Ge is the Study Director of Therapeutic Antibody Discovery and Engineering at the Shanghai Model Organisms Center. She earned her PhD from the Institut Pasteur of Shanghai, University of Chinese Academy of Sciences, where her research advanced the field of vaccine development by demonstrating that mRNA LNP platforms can overcome the limitations of traditional subunit vaccines, driving both potent neutralizing antibody responses and strong cytotoxic T-cell immunity. Prior to joining SMOC, she held a scientific position at Simcere Pharmaceutical Group. Dr. Ge's work focuses on vaccine immunology and antibody discovery with deep practical expertise in generating antibodies against challenging multi-transmembrane targets. Now, please type any questions for our speaker and panelists into the chat, and we'll address them in a live Q&A session after the presentation. So please stick around until the end of the webinar. Dr. Ge, I'm excited to learn from you today. Please take it away.
Uh, Thanks for the introduction, Alex. And hello, everyone. This is Nini. I'm a study director from pharmacology department of SMOC, and I'm responsible for antibody discovery. I'm very happy to have this opportunity to introduce the humanized antibody mice, SmocMab, and share our antibody discovery works on this mice strain. And my topic is SmocMab, a mechanism-aware platform for next-generation therapeutic antibody discovery. I will cover the following four sections: a brief overview of antibody discovery, and introduce to fully humanized antibody mouse, SmocMab, and two antibody discovery cases, including PD-L1 functional antibody discovery and a KO strategy targeting multi-transmembrane and highly homologous antigens SIP1. First, I'd like to briefly introduce our company. Founded in 2000, SmocMab specialized in the generation of mouse models. To date, we have established more than 18,000 mouse strains, over 11,000 of which have been fully validated and are available for relevant research. And in addition, we generated more than 4,000 new mouse strains each year. And we also offer customized mouse model generation services, including knockout, knock-in, point mutation, targeted overexpression, and random transgenesis. And we also maintain ready-to-use mouse strains for various pharmacodynamic models, such as target humanized mice, immunodeficiency mice, and fluorescent report mice, and so on. Leveraging our extensive library of mouse strains, we have established a full spectrum of pharmacodynamic models and provide related research services. And the SmocMab mice are the outcome of our 4-year R&D program, with which we have launched our uh, de- uh, delic- dedicated antibody discovery service. Antibody service mainly falls into three categories: in vivo discovery, in vitro discovery, and in silico discovery. Hybridoma technology is the earliest antibody screening platform, and the most early therapeutic antibodies were generated by immunizing mice, followed by hybridoma screening. But due to the sequence discrepancies between murine and human antibodies, murine antibodies always exhibit high immunogenicity when applied as therapeutics, which leads to reduced efficacy and safety risks. Accordingly, humanization engineering is necessary for mouse-derived antibodies. Subsequently, the evidence of phage display enabled the construction of human B-cell libraries for screening fully human binding antibodies. However, antibodies obtained via this method frequently possess suboptimal affinity, requiring additional affinity maturation and engineering. And the emergence of fully human antibody mice has opened new avenues for therapeutic antibody development. They generate antibodies with 100% human germline sequences while retaining the native in vivo affinity maturation mechanism for screening high quality binders. Today, the rapid advancement of computational and artificial intelligence technology is evident to all. Also, computational antibody discovery is still in its infancy. It is poised to become an indispensable tool in the near future. In summary, in vivo discovery, in vivo antibody discovery remains the mainstream workflow at present, whereas the phage display dominates in vitro affinity optimization. And a wide array of technical platforms is now available. And with that in mind, let's see which discovery platform have produced the FDA-approved antibodies therapeutics to date. And shown here are antibodies therapeutics approved by the FDA over the past 5 years. Among them, about 83% were derived from in vivo immunization, including wild-type mouse immunization, followed by humanization, engineering, and fully human antibody mice immunization. It is clear that the majority of approved antibody drugs rely on in vivo immunization paired with the endogenous affinity maturation and selection. Furthermore, the heavy chain variable regions of antibodies generated from transgenic mice predominantly utilize VH3 and VH4 germline families. And now, a variety of human antibody transgenic mouse strains are currently available. They differ in their construction strategies and the selection of human variable region gene segments, and all have been applied to varying degrees in therapeutic antibody development. At this point, you may be wondering, why do we still need the SmocMab mouse platform? And as shown in the table, developers of humanized mice have long sought to incorporate larger repertoires of variable region segments to maximize antibody sequence diversity. But is a greater number of variable gene segments always better? As mentioned, the majority of the FDA approved fully human antibodies utilize VH3 and VH4 germline genes. And the graph on the right illustrates the utilization frequency of VH germlines across the different human antibody mice platform in antibody discovery programs. It clearly demonstrated that variable genes are not equally utilized. VH3 and VH4 preferentially recruited during the natural immunization in humanized transgenic mice. And in addition, there also exists a distinct germline bias in the generation of functional antibodies. For instance, VH323 is optimal for neutralizing antibodies, but poor for agnostic activity. Therefore, the industry lacks a human antibody mouse platform engineered with functional performance and drug developability as the core design principles. This is the core motivation behind the development of our SmocMab mice. And we adopted an in situ replacement strategy. The endogenous VDJ regions of mouse heavy chain and the light kappa chain were knocked out, and the human VDJ segments were inserted at the corresponding loci. The native mouse constant region was retained to preserve the natural antibody class switching mechanism. For variable region selection, instead of incorporating all human V segments, we designed a platform centering on antibody functionality and drug developability. Taking the heavy chain as an example, we selected the 23 V segments predominantly from the VH3 and the VH4 germline families to balance the sequence diversity and developability. And once we obtain the variable region sequences with the specific target binding capacity, we can recombinantly fuse these V sequences with human IgG constant region, yielding fully human antibodies. Following the generation of the SmocMab, we first validated whether their B cell development proceeds normally. The spleen size of the SmocMab is comparable to that of wild-type mice, and moreover, the proportions of various immune cell subsets in the spleen remained unchanged, and the rates of pro-B, pre-B, metro-B, memory B, and plasma B cells throughout B cell development show no significant differences from wild-type controls. And these results confirm unimpaired B cell development in SmocMab. Subsequently, we performed high throughput B cell sequencing on splenocytes isolated from SmocMab. The data revealed that all engineered human V segments are efficiently utilized. When analyzing heavy chain CDR3 sequences, their length distribution closely matches that of human native CDR3 repertoires. Analysis of amino acid usage frequency within CDR3 regions confirmed functional somatic hypermutation, which is an essential process for generating broad sequence diversity and high affinity antibodies. Over the past year, we have performed immunization with a wide range of antigens on SmocMab. As shown in the data, antigens of varying molecular weights all elicit robust antigen-specific antibody responses. Even when primary immunization is started at 16 weeks of age, the mice can induce potent antigen antibody responses as well. Moreover, it is widely accepted that biopsy mice generated stronger humoral responses than Black 6 strains. The three cases below show that Black 6 and the biopsy mice can each outperform one another depending on the antigen used. And in this case, we can see Black 6 6-based SmocMab mice achieved higher antibody titers than biopsy mice. Overall, our SmocMab platform delivers outstanding antibody response performance. Once serum antibody titers are qualified, antibody screening can be started, and there are three mainstream screening workflows: hybridomer technology, phage display technology, and single B sorting technology, each with distinct advantages and limitations. Hybridomer technology is the earliest and most classical screening approach. Functional screening can be integrated directly into the selection workflow. Its drawbacks include an extremely heavy workload, high randomness, and low efficiency during fusion with myeloma cells. The viability of post-fusion cells also directly impact the recovery rate of positive antibody clones. And the full screening cycle usually takes more than 3 months. Phage display enables multiple rounds of positive and negative panning, making it ideal for projects requiring negative screening. However, this technical disrupts the native heavy and light chain pairing of B cells during the library construction, which may lead to poor expression issues with isolated antibodies. Single B sorting offers a more streamlined workflow. Antibody positive B cells are isolated via sorting, followed by direct sequencing of individual cells to obtain naturally paired heavy and light chain sequences. This method therefore delivers extremely rapid screening turnaround. And antigen-specific B cells can be divided into memory B cells and plasma B cells. So accordingly, single B cell sorting is further categorized into memory B cell sorting and plasma B cell sorting. As we see, B cell director in the bone marrow and complete the BDG recombination prior to maturation. After encountering antigen stimulation in the lymph nodes and the spleen, they undergo somatic hypermutation and class switching to generate and select high affinity antibodies. A subset of activated B cells differentiates into memory B cells to allow rapid secondary immune responses upon re-exposure, while another subset becomes short-lived plasma cells to clear ongoing antigenic challenges or infections. Activated B cells always exhibited a spectrum of binding affinities. They migrate into the germinal centers where TFH TFH cell-dependent antigen presentation mediates the selection of high-affinity B cells and delivers vital signals. Researchers have demonstrated that high-affinity B cells are enriched within the germinal center and differentiate into the plasma B cells. In contrast, the antigen binding affinities of memory B cells failed to support further clonal expansion in the GC and retained clones with a broad spectrum of antibodies. Accordingly, we concluded that plasma B cell produce antibodies with superior affinity compared to memory B cells. Our single B cell platform uses droplet microfluidic technology. We encapsulate enriched plasma B cells fluorescently labeled anti-mouse Fc antibodies and fluorescently tagged antigens or overexpressing cell lines within the individual droplets. If a plasma cell secretes antigen-specific antibodies, the fluorescent signal from the secondary antibody accumulates inside the droplet and co-localizes with the antigen fluorescence signals like this. And the droplets containing antigen-specific plasma cells are then sorted for sequencing. The full workflow consists of B cell preparation, droplet generation, incubation, and sorting, which takes around 6.5 hours in total. The subsequent single B cell sequencing requires an additional 2 weeks. Compared with the hybridoma and the feature display workflows, this approach delivers substantial faster screening. Next, I would like to share our antibody discovery case study targeting PD-L1. Following the sorting workflow described above, we isolated over a dozen high-affinity antibodies that bind human PD-L1, and the SPR results showed that nearly half of these antibodies have comparable affinities to the benchmarker, and we then assessed their cross-reactivity with the homologous protein, and the data showed that 9 out of these 12 antibodies maintained strong binding affinities to the homologous autolog. We further characterized the functional activity of these antibodies, including their endocytosis and blocking abilities. The results showed that 9 out of 12 antibodies exhibit comparable internalization to the benchmarker, and 4 out of the 12 antibodies exhibit comparable blocking activity to the benchmarker. Based on the functional characterization data, we selected 5 lead antibodies for in vivo pharmacodynamic evaluation. Among them, AB126 only induced endocytosis without blocking activity, while the other 4 antibodies possess both endocytic and blocking functions. In vivo monotherapy results demonstrated that therapeutic efficacy primarily relies on the PD-1, PD-L1 blocking activity, and the AB16 and the AB17 exhibits anti-tumor activity comparable to the benchmarker. Notably, no variable region sequence engineering was performed on these antibodies prior to in vivo testing. So their potent in vivo performance matches the benchmarker drug demonstrates that antibodies directly generated from SmocMab of high quality. The second case study I will present covers our antibody discovery solution for the challenging target STEAP1. STEAP1 is a sixth transmembrane protein highly expressed on the prostate cancer cells. Studies have revealed that STEAP1 overexpression correlates with aggressive disease progression and poor prognosis in metastatic castration-resistant prostate cancer. Xaluritamig has demonstrated promising therapeutic efficacy in clinical trials, confirming STEAP1 as a high potential target for prostate cancer treatment. However, antibody development against this target poses substantial technical challenges. The first major challenge lies in eliciting robust antibody responses against this target. As human and mouse, STEAP1 share about 84 sequence homology, which leads to partial immune tolerance, and additionally, its limited extracellular regions further hinder effective immune reactivation. To tackle targets of this type, we have developed a knockout-based immunization strategy. We delete the STEAP1 genes on the SmocMab to break central immune tolerance, and the serum titer data shows that STEAP1 KO SmocMab mice can elicit much higher antibody titers compared to the wild-type mice. In contrast, not all wild-type mice could induce measurable, effective antigen-specific responses even after 4 immunizations. And as mentioned earlier, overexpressing cell lines can be utilized as screening materials in the product single B sorting workflow, which could overcome the expression challenges associated with multi-transmembrane targets. And we obtained a total of 2,794 antibody sequences via single B sorting, and these antibodies can be mapped into 650 clonal types. The VDJ usage profile reveals that these sequences are predominantly derived from BH3 and BH4 germlines. Analysis of heavy chain of CDR3 length shows a broad range of lengths and abundant clonal diversity, laying a solid foundation for generating antibodies with diverse functional activities against STEAP1. After sequences analyzing, we have selected 51 candidates for recombinant expression and purification, followed by binding characterization. And the majority of these antibodies exhibited a stronger cell binding affinity than the benchmarker. And notably, nearly 78% of these antibodies demonstrated superior binding intensity relative to the benchmarker when tested on the lean-type tumor cells. And we further assessed the endocytic activity of these antibodies. Antibodies highlighted in yellow exhibit endocytosis rates exceeding the benchmarker, while those marked in blue show the detectable but weaker internalization. And a subset of clones like AB11 and AB44 exhibited no measurable endocytosis at all. Based on their distinct function profiles, these antibodies can be deployed for divergent therapeutic modalities. For example, clones with potent endocytosis are ideal for ADC development, whereas binder selection endocytic activity can be engineered for TCE programs. In conclusion, we have established a high-performance, fully human antibody discovery platform based on SmocMab, which is rationally designed focusing on antibody functionality and clinical developability. Combining the antigen KO strategy with microfluidics-based single B-cell sorting platform, we enable ultra-fast and efficient discovery of natural-paired functional antibodies, even against the challenging targets such as multi-transmembrane domains and highly homologous antigens. That's all. Thank you for your attention.
Great. Thank you so much, Ning Ning. That was a fantastic presentation. And now we've got a couple comments and questions coming into the webinar chat. So if you could stop your screen share so we can see the entire panel, that would be the best way to start. And if we need to refer to some data, you can share that again later. Great. So the first question we have is, regardless of the antigen, how does the immune regimen impact antibody response and clone diversity?
That's a good question. Immunization strategy do influence antibody responses, and to preserve clone diversity, the immunization times should be limited, as clone diversity will be reduced after too many immunizations. So antigen also should be rationally designed to improve its immunogenicity and to avoid too many immunizations.
Great, thank you. And I want to open this up to the panel too. So if anyone else here wants to chime in on any points, please feel free. Next up is, well, on the idea of antigen format for the SmocMab mice. What format is preferred for the immunization, and is there any bias for different types of programs?
Like wild type mice, SmocMab can produce potent immune responses against diverse antigen formats, such as protein, VLP, and mRNA, all of which works well.
Great. Next question here is, so how, if you need to generate a target knockout SmocMab mouse, what's the time frame to get that new model put together to start the immunization?
Pardon?
If you need to generate a target knockout SmocMab mouse for before immunization begins, what's the timeline and the lead time for preparing that knockout?
It will take 3 months from construction to be ready for immunization.
Okay, great.
Yeah, I want to mention that we generate the target knockout mice directly from our SmocMab mice. We are not crossing our SmocMab mice with knockout wild-type mice. So it saves at least 1 year to get the target mice for immunization. This is the advantage of our platform.
Great, thank you. All right, next up, we're going to move more into kind of the technical nuts and bolts of how we do the sorting. So obviously, you talked about several different technologies. Can you talk a little bit more about the range of alternate technologies for single B cell sorting other than microfluidics?
Yeah, I think Alex, that's a great question. I think Union presented for single B cells. There are 2 major technical routes. One is the memory B cells. Usually people do FACS sorting. the other way is what we did is a microfluidic based plasma cell sorting. And Ningning presented the advantage of the plasma B cells, its affinity optimized antibody discovery. So each technology has its own advantage. And in the field, there are, the scientists are debating on which is more productive and which is more ideal for therapeutic antibody discovery. To me, the flow cytometry-based, single B cell sorting has one shortcoming. You need to have a soluble antigen. For difficult targets like STEAP1 Ningning presented, there's no way for us to do fact-based sorting. Technically, you can use VLP to do fact sorting, but the background will be much higher than the microfluidic-based single B-cell antibody discovery. There are also debates on the developability of antibody discovered using fact-based sorting versus microfluidic-based sorting. And the current common knowledge is plasma B-cell derived antibody is better developability-wise.
Great, thank you. Actually, on the same topic of different sorting technologies, so a lot of people are familiar with the beacon technology. Could you explain the difference between the microfluidic approach and the beacon?
Yeah, I think the major difference is on the throughput. For beacon, it's a chip-based. Each chip, there are 14,000 wells. They call it pens, 14,000 pens. And each instrument, you can use 4 chips. In general, we only do 2 because the longer time you put the plasma cell on beacon, the poor the quality of the isolated single B cells. And that's to say, for a beacon experiment, the maximal diversity you can put into the equipment is 56,000 B cells. For microfluidics-based approach, like the instrument we are using, we can easily put millions of plasma B cells into the instrument. Yeah.
There's orders of magnitude difference. That's crazy. Actually, another question about numbers. How many mice do you need to immunize to get ready for one sorting run where you're putting millions of B cells into the microfluidic sorter?
It's a really practical question. Usually, one mouse can be performed for one sorting run, but if we want to get more sequences, we recommended to use 3 or more mice for a sorting run. So we can do high throughput PCR sequencing and get more sequences.
Okay. When you have multiple mice, what is the percentage of overlap in the B cell receptor pool between the animals? Like, how much more diversity do you get with every successive animal you add into the experiment?
We can get hundreds or thousands of antibody sequences in one sorting run, and there must be some repeated sequences and a little of IgM sequences. So we needed to perform sequence analyzing, and we always choose 30 to 40 unique antibodies for expression and binding verification, and the positive rate usually range 40 to 90%.
Okay, thank you. And then another kind of technology-based question is how do we combine the antibody humanized mice with the advent of new in silico and AI antibody drug discovery techniques? Like, just looking forward into the future, how do we combine those technologies?
Yeah, I think that's a great question. We are looking for partners to combine these 2 technologies together. From the case study Ningning just presented for PD-L1 and STEAP1, in each antibody discovery campaigns, we isolated over 6,000 different antibodies. I think in the future, in the project, because we are not a drug discovery company, we are a contract research organization. In this case, we only pick a couple dozen to produce and to characterize. It's always debating which one is the best one and did we miss the best antibody. If there's an AI capability, we can work together to understand more the depth of the immune response and how to quickly, efficiently generate a develop a therapeutic antibody.
So more of like a screening tool and an optimization tool for after we've developed like a repertoire of antibodies.
Yeah.
Fantastic.
Yeah, I think definitely in this field is very dynamic and people challenge also use AI completely wipe out all those white lab or in vivo immunization. Our AI heavily rely on in vivo immunization data to train them or to work together to improve this field. Some people could believe that because AI and need more like immunization, humanized antibody, humanized mice. So that's a kind of a very big open question. So I think audience also want to get some insight. For my own experience or perspective, I think AI is a great tool, but nowadays they cannot generate real new knowledge. They only try to organize or summarize the existing knowledge. Definitely there's a machine learning, but learning also from the raw data. I think the immunization and the big antibody pool will provide the very strong and very necessary to improve the AI. I think AI and wet lab will like walking in two legs, and that's my personal perspective. I don't know other people.
Yeah, I think that's a fantastic. So for our campaigns, we generate a couple thousand antibody sequences, and the majority of them are binders, even though the binding affinity varies. So we have higher quality data sets to enable AI model development. We not only have a positive data, we also have negative data. I think a lot of AI machine learning is based on the public available sequences, and the quality is not there. Here we have a high quality antibody discovery data. Not only binding, we also have a in silico developability data and functional data.
Yeah, so follow Dr. Chen and Dr. Jin's answers. I think whatever the AI can design antibody sequence de novo or from big data, definitely you need some wet lab to verify the sequence of antibody, the function, PK, whatever. Because we have all the fully human, we have the target human and mouse model, we can test the in vivo efficacy, maybe some on-target, off-target toxicity of the antibodies, no matter it's from the AI or from our platform. All those data accumulated together, we can train the AI system to design new antibodies with high efficacy, low side effects, and more druggability in the future. So I think this kind of the basement to build the AI building in the future.
Great. And now we have another question about, the binders that we get. How frequently do we get an antibody that cross-reacts between human, cyno, and mouse? Is that like a serious needle in the haystack, or is this something that happens, with some frequency?
it really depends on the target.
Yeah.
For some target, it's much easier, and for some targets, it's very difficult. To give you an example, for CD3 antibody, that is widely used by everybody to make T-cell engagers. For CD3 antibody, it's impossible to generate a functional human cyno mouse cross-reactive antibody. So there are several ways to go right now. Some people use a mouse surrogate just to bridge the study. And, the most popular anti-CD3 antibody currently in the clinic is SP34 humanization variant. It's only human cyno cross-reactive. So I think for SMOC, our strength is the transgenic mouse models. Even there's no human mouse cross-reactivity, we have a target humanized mice to enable the downstream in vivo pharmacology study.
Great.
Yeah, that's exactly. I think first, I think we can, if we knock out the antigen from our SmocMab, the animal, the chance to get cr cross-species antibody is higher.
Yeah,
Second,
right.
we don't sacrifice the good binder, good antibody because cross activity. If you only bind the human one, I think our thousands uh, therapeutic target humanized mice can help you. Not only us. I think in this field is relatively new and like 5 or 6 years, but grow rapidly and almost all the company accepted this idea. But for cyno and mice, cyno and human, the chance is very high, right?
Yeah.
Do we have some statistics like more than 95% the cross-reactive or?
Yeah, in general, for human and cyno, the homology is way much higher than human and mouse.
Yeah.
Yeah, so far we have validated our platform with three antigens, the PD1, PDR1, the STIP1, and also another one we tried is CD71. And from all those three projects, we identified both human and cyno cross-reaction antibodies at the same affinity level. Pretty good data. I think, as James said, because we knock out the target gene, and so it's very easy for the animal to develop the antibodies against whatever epitope. Because for some functional antigen, so the functional domain of the antigen normally is very conservative. So if the human mouse cyno are very similarity is very high, sometimes it's really hard to develop antibody against that epitope. If we knock out the gene, it's kind of a naive antibody, a naive antigen. It's very easy to get the antibodies targeting every epitope.
Great. So that was the last question we have in the webinar chat. And I want to open it up to the panel for any other remarks or thoughts or other insights to share.
Yeah, we just talked about the target knockout. To me, rather than manipulating immunogen, the different immunization schemes, the target knockout is a very effective way to break immune tolerance, especially for the target is of a high human mouse homology. But there's one thing to be aware of. If the target is involved in immune responses, the target KO, you might completely eliminate the mouse immune response. Just keep this in mind.
Yeah, so sometimes knockout of the target gene might be lethal. That's why we also have another approach that we can introduce permutation in the specific epitope that you want to develop antibody against. So with a permutation, the mice can survive and you can still immunize the mice with the wild type antigen. So it's still have the opportunity to develop antibodies against this wild type antigen. There's another option if you cannot knock out the whole gene of the antigen.
So a little bit business promotion. As you know, I'm the business person, and but by training, I was also scientist. Uh, so we have a very flexible business model. You can just pay for services, and we will remove those downstream legal entitlement for your IP products, and that's our new business model. And also we provide how difficult the target it is, as long as we evaluate it and we evaluate the risk, we will provide a 100% guarantee. And we are quite sure because we are so confident with our platform and technology, and we will take all the risk on our own. We will not put you on any risk. So any questions from audience?
Yeah, I already answered a question from one of the audience, and he asked if we can do nanobody from this platform. Actually, this is for regular human IgG molecule generation, but we have other mouse that can generate human nanobodies and also common light chain IgG for bispecific antibody assembling. So if you are interested in our platforms, feel free to contact our BD, our title BD managers. Then we can provide more information and talk about your project with a CDA in place.
And also, we built our capability to generate nanobody mice or VAHH using llama or alpaca immunization. So it's fully available here.
Okay, there are two more open questions we didn't answer, right?
Really?
Have you
Okay.
ever considered how mass spectrometry could accelerate your antibody development?
Yeah,
I totally don't know.
yeah, that is a great question. I think it's trending up right now using mass spec to do antibody discovery. Basically, you just try to deconvolute what's available in the serum and discover antibody on a protein level. We will be excited to collaborate with the industry academic partners to try this out. From my limited experience, the sensitivity of the methodology is critical to discover those rare antibodies. But for the dominant immune response, the antibody against the dominant epitope, this method is working right now.
Uh, okay, and there's another one very simple question. When are we, for us, available to talk? I think we are available all the time. Yeah, I'm in Boston, and Lei in Boston, and Alex in California, and Dongxiao in Texas, and Ningning is in China. And whatever your time zone, you always can find us. And welcome to shoot an email or other find our LinkedIn, and we will address your question anytime.
Great.
Yeah,
Well,
also, we are building up the same antibody screening equipment in Houston, Texas right now. So if you think your target is sensitive, you don't want the antibody to be screened in outside the United States, we can do the service here in Houston, Texas. We have the similar equipment. We have same equipment as Shanghai. We have.
Okay, so another will provide a recorded video. Yes, we provide and follow our LinkedIn and find our Southern model organism LinkedIn, and we will publish all those information on our LinkedIn.
And we'll send a link to the recording to every attendee after the presentation today.
Okay, even more convenient. But follow LinkedIn is we appreciate.
Yes. Great. Well, I think with that, unless there's any other burning questions from the audience, we will conclude today's webinar. Thank you all so much for joining us. It's been an absolute pleasure to get to share with you more about our antibody discovery platform. Oh, I have a hand raised. I will hold off on the salutation until we get the question. Oh.
Okay.
All right, we'll discuss this later. But anyway, thank you all so much, and thank you, Dr. Get, for your presentation. This was fantastic, and we look forward to working with all of you in the audience on your next antibody discovery project. So everyone have a great rest of your day, and we look forward to speaking further in the future. Thank you very much.